In the previous presentation on carbohydrate catabolism, we noted that being able to do aerobic or anaerobic respiration requires quite a bit from an organism. It requires that the organism be able to make acetyl-CoA. It requires that the organism have a Krebs cycle. And it requires that the organism have an electron transport chain, either an anaerobic or an aerobic one. It also requires that if it's an aerobic electron transport chain, that there be oxygen to serve as the terminal electron acceptor. If there's an anaerobic electron transport chain, well, then there has to be a non-oxygen inorganic molecules such as sulfate to serve as the terminal electron acceptor. Well, what if none of those conditions can be met? How does an organism regenerate the NAD that it used in glycolysis? How does it get that back? This brings us to fermentation, which does not use the Krebs cycle. It does not use an electron transport chain. So now we're going to be talking about what's happening on the right side of our figure, the left panel of that right side figure. What we're going to do in fermentation is we're going to use an organic molecule such as pyruvate, pyruvic acid, or a derivative of it as the final electron acceptor, as the terminal electron acceptor. So fermentation, like glycolysis, can occur under anaerobic conditions, that is, where there's no oxygen. And we're going to use fermentation to regenerate our NAD. So what fermentation does in a nutshell is it provides cells a way to regenerate NAD without there being oxygen or without there being another inorganic molecule to serve as the terminal electron acceptor. In fermentation, organic molecules such as pyruvic acid or a derivative of pyruvic acid are going to be the terminal electron acceptors. And when we do fermentation, if we look at the right side of our figure, what we see is that in fermentation, that NADH that was generated in glycolysis is going to be oxidized to give us back our NAD, and we're going to dump the electrons, the hydrogen ions and the associated electrons, onto pyruvic acid or a derivative. So we're going to reduce the organic molecule. To reiterate, the essential reason for being, the essential function of fermentation is to regenerate our NAD so that we can continue to do glycolysis, because recall that cells have a finite, a limited pool of NAD, and if we use it all up, that is, if we make NADH out of it, then we can't continue glycolysis and we can't make any ATP, and that's usually lethal. However, to note about fermentation is that it's energetically inefficient, as most of the potential energy, most of the ATP making energy, is going to remain in the bonds of the fermentation products. In addition, fermentation products are toxic wastes to the cells that make them. So typically, cells that are obligate fermenters. That is, the only way they can get their NAD back is through fermentation because they, say, lack a Krebs cycle, they lack an electron transport chain. Those are going to grow far more slowly for two reasons. One, they get fewer molecules of ATP per glucose, and because they're poisoning themselves. If you've noted over recent years, the alcohol content of products like wine has steadily increased. Why? Because what vintners have done is develop strains of yeast that can survive higher levels of alcohol. That is, they've created strains of yeast that can survive higher levels of their own waste products. So it's sort of like if we created humans or some other animal that could wallow in our own filth and survive better. Now, another thing to note is that many fermentation products are useful to humans. Things like ethanol, which of course is the drinking alcohol, acetic acid, vinegar, butanol, and there's a red star by that one, and we'll talk about it some more, and then lactic acid that we find in yogurt. We noted earlier that in fermentation, pyruvic acid, or a derivative of pyruvic acid, was used as the terminal electron acceptor. And so what we're looking at in our figure is a diagram of a variety of fermentation pathways and then the microbes that use those pathways. Now, don't panic. We're not expected to learn all of these different fermentation pathways or the different microbes that use them. This is just to illustrate, again, that different microbes use different fermentation pathways, and it turns out that an organism cannot have every single metabolic pathway there is. We've seen that in that humans don't have anaerobic respiration because we don't have the components for it. So, for instance, lactic acid bacteria, such as lactobacillus, would not be able to do, say, the same fermentation that acetolactic bacteria would do. So what I'm getting at is that we can use an organism's metabolic capabilities or lack thereof as part of our profiling kit. That is, we can use an organism's ability to, say, produce lactic acid, but not propionic acid, as a way of distinguishing microorganisms, and that's something that we do in the lab. We noted also earlier that many fermentation products are useful to humans, so many important substances are produced by fermentation. And our figure shows the organisms that do certain fermentations and the products that are made. For instance, Propionibacterium does the propionic acid fermentation, and that's used for making Swiss cheese. So the carbon dioxide that's produced in the fermentation makes the holes in the Swiss cheese. The propionic acid gives the Swiss cheese its characteristic flavor and ammonia. Yet other bacteria, such as Lactobacillus and Streptococcus and Aspergillus, do a lactic acid fermentation and so produce such foodstuffs as cheddar cheese, yogurt, and soy sauce. Yet another microbe, the yeast, the fungus, Saccharomyces, does the yeast alcoholic fermentation, which produces carbon dioxide and ethanol, which we find in wine, beer, and liquor. We'll save the clostridial fermentations until last. We also have bacteria that ferment to give acetic acid. So E. coli, Escherichia, and Acetobacter ferment and give acetic acid to produce vinegar. Well, now, what about Clostridium? Well, it turns out that the Clostridia, that's members of genus Clostridium, are among the master fermenters of the microbial world. And as such, they have been important historically. So during World War II, the Nazis had managed to gain control of the oil fields in the Middle East. And petroleum is needed for a number of products, such as fuel and cordite. Cordite is what is used to fire guns. And so the Allies, especially the British, needed to get an alternative source of the material for making cordite. Enter Chaim Weizmann, who was a British Jewish scientist who held the patents on the acetone butanol fermentation that we see in the green part under Clostridium. And he was approached by members of the government as to whether these patents could be used for patriotic purposes to help in the war effort. And he said, sure, I'm a patriotic person. I want to help my country. But as a Jewish person, I'm very concerned about what's happening to my people in Europe, where now the Nazis were rounding people up and shipping them off to concentration camps and killing them. And so his price for the use of his patents was that there be a state of Israel. And so Weizmann became the first president of Israel, and the price was the use of these acetone butanol fermentation pathways, which Weizmann had patented the best ways to do those. Amazing what microbes can do. So let's put our knowledge about fermentation into practice with some critical thinking. So all E. coli, whether it's E. coli like we have in our intestines, or whether it's E. coli 0157H7, the cause of foodborne illness in many cases, or various other varieties of E. coli, well, they're E. coli. They all look alike in a microscope. They're all gram-negative short bacilli. So how can we determine whether we have E. coli 0157 versus just regular E. coli? And our photo here is that of E. coli 0157H7, the pathogen, in purple, binding to the microvilli of the intestine in yellow. And normally microvilli would have a dome-shaped top. But what's happening is that the E. coli 0157H7 secrete a molecule that causes the cytoskeleton of the microvilli to change shape to form that flat top, the pedestal, so that the E. coli 0157H7 can adhere better and then have its wicked ways with us. So our question, all E. coli look alike through a microscope. So how can we differentiate E. coli 0157 from other E. colis? Well, we can do it by examining its ability to ferment different sugars. And what it turns out is that E. coli 0157 can't ferment sorbitol, whereas other E. coli strains can do this. So we can see this in our bottom panel. If we look to the tube at the left, we can see that it's yellow, and we can also see that there's gas in the little inverted tube. And the medium that we're using is called PR for phenol red, which is a pH indicator, and then it's got sorbitol as the sugar. So our little friend E. coli at the left can ferment sorbitol and produce acid and gas. So it's going to be yellow with gas in the little tube. That's positive. If we look to the right, we see E. coli 0157 growing in the tube, and the medium stays red because it's not fermenting the sorbitol to produce acid, and there's no gas in the little tube because we're not producing any gas either. So E. coli 0157 gives us a negative result, the red. So this is how we can use an organism's metabolic capability, or in this case, lack thereof, to profile it, to get a handle on what it actually is.